Now, I would like to talk about these drugs. So, this methotrexate is an anti-cancer drug. Likewise, azacitidine, it's a non-standard amino acid which is also utilized as anti-cancer as well as antibiotic drug. Then diazonordleucin, this is also one non-standard amino acid, and this is also used as anti-tumor agent, that is same anti-cancer agent. About 6 mercaptopurine, it is also an anti-cancer drug used for leukemia and autoimmune diseases also. Now, mycophenolic acid, which reversibly inhibits IMP dehydrogenase, that is also used as one immunosuppressant drug. So, again the overview of the entire pathway. Now, I hope you are more comfortable about this purine synthesis. This for you, common sense is in an uncommon degree is what the world calls wisdom. Now, coming to the second pathway, we have spoken about de novo synthesis. Now, the salvage pathway, where actually whatever is left after some devastation, when we recover that, that is known as salvage. So, be it like I have shown here the sea breakage, usually for recovering the remnants or whatever possible from sea breakage is known as salvage, but from some burning of certain houses or something, if you can recover. So, the same thing here. Similarly, we recover the nucleotides and the intermediates during their catabolism and utilize them as the precursor of newer nucleotides. So, that is why it is known as salvage pathway, and why it is really required. So, it provides purine to tissues that are incapable to produce purine by de novo pathway. So, like human brain, it has very less PRPP aminotransferase, and RBC, WBC, they cannot synthesize 5-phosphoribosylamine. And as per nature's rule, it also economizes the energy expenditure. Hence, the salvage pathway is very important. So, these mononucleotides or the intermediate nucleotide catabolism intermediary products, they are used either by phosphorybosylation of purine bases or phosphorybosylation of purine nucleosides. So, how it is done? Coming to phosphorybosylation of purine bases. So, already purine is there, suppose it is adenine, we add PRPP and the enzyme is adenine phosphoribosyl transferase. So, what we transfer here is the phosphoribosyl group to this adenine, and we get adenosine monophosphate. Similarly, from hypoxanthine, hypoxanthine is just the last but one product in purine catabolism. So, in presence of hypoxanthine guanine phosphoribosyl transferase, a big name, you can remember it as HGPRTase. Okay, the PRPP transfers the ribosyl and phosphoribosyl group and inosine monophosphate is obtained. So once we get IMP, we can either produce AMP or GMP, so no hassles. The same HG PRTase also converts guanine to GMP. Fine. So just remember this, because later on we will come across some disease where this goes defective. The other method is phosphorylation of purine nucleoside. So the purine nucleosides are still intact, they are not dissociated into sugar and nitrogenous base. To that, in presence of ATP, a phosphate group is transferred in presence of kinase enzyme, and finally we get this PURP plus ADP. So further we can synthesize desirable nucleotides. The first product that is produced actually are ribonucleotides. To make it deoxyribonucleotides, where they will go? They will go to DNA. Fine. So it is done by reduction of ribonucleotide diphosphates, and this enzyme complex is usually active during cell division, where the DNA is just about to be duplicated. So just before S phase, this enzyme complex is active, and this reduction requires thioredoxin reductase and NADPH. Now if you remember, thioredoxin and thioredoxin reductase, they contain the 21st amino acid selenocysteine. I hope you remember the 21st amino acid. So not in many reactions NADPH is used. So this is one of those important steps or metabolic pathways where actually NADPH is being used. Now this ribonucleoside diphosphate is converted to 2' or 2' deoxyribonucleoside diphosphate, which is meant for DNA alone. In presence of ribonucleotide reductase, in due course reduced thioredoxin will be converted to oxidized thioredoxin, and again it will be obtained back with the help of thioredoxin reductase, and for this step NADPH is required. So this is what is the pathway, salvage pathway, and the production of deoxyribonucleotides. So with this we come to the end of our lecture on purine synthesis. You can find this PowerPoint on my website vpacharya.com, and also this video on my YouTube channel. Thank you. We will meet for the next class, that is pyrimidine synthesis. Bye bye till then.